Kia ora koutou, my name's John Hosking, I'm Dean of Science here at the University of Auckland. It's my very great pleasure to welcome you to the inaugural lecture of Karen Waldie. Karen was born in Victoria, BC, Canada, and received her BSc at the University of Victoria, that's the BC one. She gained an MSc and PhD at the University of Calgary, where she met her Kiwi husband. They moved to New Zealand, and Karen worked with the world-famous Dunedin study before joining us here in Auckland in the School of Psychology and the Centre for Brain Research. Karen's research interests can be broadly categorised into those that rely on acute cross-sectional experimental techniques and those which rely on data collected from longitudinal studies. Her experimental work combines techniques from cognitive neuroscience, such as functional magnetic resonance imaging, or fMRI, and EEG, with clinical neuropsychology. This is designed to make deans trip up as they read words in it clearly. For example, she's been able to show brain-based differences between neurotypicals and individuals with dyslexia. This helped the Ministry of Education to formally recognise dyslexia in 2008. Seems like an incredibly short time ago. She's currently leading a large fMRI study to determine the neural basis of dyslexia, dyscalculia, and ADHD. Her involvement with national longitudinal studies, the Dunedin study, Auckland Birthright collaborative study, and growing up in New Zealand, reflects her interests in lifespan development, as well as the precursors and determinants of neurodevelopmental disorders. In simple terms, as Karen put it when she was interviewed for our local rag, Karen studies the predictors and causes of problem behaviour and cognitive problems in kids. Karen's going to tell us more about this in her talk this evening. So please join me in welcoming Professor Karen Waldie.
Thank you so much. Thank you, John. Tēnā tātou katoa. I'd like to begin by spending a few minutes explaining my title, Genes, Brains, and Neurodiversity. What I mean by neurodiversity includes these developmental disorders: dyslexia, dyscalculia, attention deficit hyperactivity disorder, and autism spectrum disorder. I'm interested in the cognitive and biological markers of these conditions, and here I list the main cognitive impairment for each. Now, I just want to emphasize that these are only considered disorders because our modern society values literacy, numeracy, sustained attention, and social skills. I have no sense of direction, but my getting lost is not considered a disability in today's society. I'm interested in comorbidity and mental health because developmental disorders rarely occur alone. And I include this figure to remind us that ASD sits on a gradient, a spectrum from low functioning to very high functioning. But this is true for all cognitive functions and dysfunctions. A diagnosis involves a relatively arbitrary categorical cutoff along a continuum of ability. And finally, developmental disorders are due to differences in brain structure and function. They're all influenced by both nature and nurture. And parents would do anything to help their kids navigate our modern society. This is I know, right? This is the list of topics that I hope to cover tonight, and they represent a chronology of the past 22 years of my research career. But before I begin, I want to make some important acknowledgements and take you through a little bit of my early life that might be a bit relevant. I'd like to acknowledge the Faculty of Science in particular for funding so much of my brain imaging research, and to Anne Gaze, thank you. and my students. And I would not be here tonight if it weren't for these brilliant minds. Notable colleagues, Mike Corbelis, Ed Mitchell, John Thompson, Sir Richard Fall, my Dunedin study group, Anna Wilson, my Minds for Minds colleagues, Susan Morton and Liz Peterson from Growing Up in New Zealand. Professor Ian Kirk is my husband and colleague, and we met in Canada through a mutual love of neuroscience, and we have produced 23 research articles together. But more importantly, we have produced this little boy, Zachary, and I want to acknowledge him for being such a good sport over the years. He has spat into tubes for genetic testing. He's been a pilot subject for brain experiments and brain training. And to my coffee group and other friends here tonight, thank you. I was born in 1968 in Vancouver, BC, and raised in Victoria, Vancouver Island. And my name is Karen, but I'm considering changing it to Karina, because 2020 has not been a good year to be a Karen. To introduce my background, I have taken a few slides from my Psych 326 course on adoption studies, because I am adopted. So my wonderful parents wanted 6 kids, but they could only have one, my sister Jody. They adopted my brother 3 years later, and then me 4 years after that. And my biological parents, Dave and Lexi, they were high school sweethearts, and they had me during their first year of university. They went on to get married and have 2 boys of their own, Drew and Reed. And I met them all in my early 20s, and I continue to have a good relationship with them. So back to my slides, and 2 points from adoption studies. The first, despite how I look here, I love my siblings. My sister and brother, in the early years, we shared the same interests and activities and characteristics. But as we got older, there were differences apparent. So for example, my sister Jody, she's a hippie domestic goddess who grows all her own food, but we still share some things in common. And secondly, my biological mother here, Lexi, we are extremely similar. She did her graduate studies in microbiology and taught at UBC for many years. And here is my beloved Mum Betty, late Mum Betty. So although she was very different from me, she was my absolute rock, and I do dedicate tonight to her. So I did my BSc at the University of Victoria, and at the same time I was a caregiver at Queen Alexandra Hospital for Children. I did my honours thesis with the late Professor Ottfried Spreen, and he was a very well-known neuropsychologist. He wrote many textbooks and neuropsych tests, and he was given a Lifetime Achievement Award at the 36th annual meeting of the INS Society in Hawaii. And here, I was there, luckily, here with Mike Corbelis and my former PhD student, Gurgitsa. For my honors thesis, I was introduced to the wonderful world of prospective longitudinal studies and they are the gold standard way that we understand and study human development. And Ottfried started this study in 1976, and he followed learning disabled children as they grew up. And back then, we used the label learning disabilities to mean reading disabilities. And I was interested in why some children with learning disabilities continued to have significant police contact at age 25. And what we found was certain characteristics, such as impulsivity and poor judgment, made these individuals more susceptible to persisting delinquency. And these characteristics go into self-control. And important research from the Dunedin study found that self-control predicts health, wealth, and public safety. For my graduate studies, I moved to Calgary, Alberta, and did my MSc and PhD with Professor Emeritus Larry Mosley. I did my MSc in the area of cognitive psychology, but for my PhD, I wanted to do something a bit different. I happened to attend a basics conference where the very famous Mike Kazanaga presented a talk on his split brain patient research. And what he told us is that the left hemisphere is dominant. It holds our oral language capabilities. And because of this, he says the right hemisphere is boring. He said that he would never go on a date with the right hemisphere. So I'm quite keen on the underdog. So I spent my PhD trying to determine what the right hemisphere can do. So I looked at hemispheric specialization for reading in typical readers of all different ages. And I also recruited 45 children with dyslexia from a specialized school called Foothills Academy. And I also became clinically qualified and spent a one-year internship at the Calgary Learning Centre. And I got married. Ian and I moved to Dunedin, New Zealand, just for 1 year, as he was working on an HRC grant at Otago. And I was finding it fairly difficult. I was fresh out of a PhD and trying and failing to get funding for my dyslexia research. And the Ministry of Education hadn't formally recognized dyslexia as a real disorder of reading at this time. Meanwhile, the Dunedin Multidisciplinary Health and Development Study was advertising for a biostatistician for their age 26 assessment. And though I had certainly heard of this world-famous study, I didn't really consider myself a statistician. But I got the job, and I have not regretted it at all. It is the richest archive the world will have on human development. And I was also introduced to the fascinating area of how early life influences, like sunlight exposure, can impact on child development. And somehow I also became a migraine expert. Here I list some of the main characteristics of migraine, and for about 3 years I wrote papers on self-perceived health status in migraineurs, the physical and psychological correlates of migraine, and the cognitive correlates of migraine. And in this paper here, my co-authors, Marcus Haussmann and Barry Milne. And we found evidence that there is something that we call a migraine personality. So migraine is related to a history of stress reactive and harm avoidance personality traits, a history of anxiety, and late onset depression. In 2001, I got a job as a lecturer at the University of Auckland, and I was fortunate to remain an associate investigator with the Dunedin study. So when Spelled New Zealand asked me to speak at their upcoming AGM, I was able to speak on the topic of how do children with dyslexia turn out years later. So earlier, Dunedin study researchers had found 7% of the cohort were identified as having serious reading impairments. And I looked at a variety of outcomes and compared these two groups. At age 26, those with dyslexia were significantly less likely to have achieved a bachelor's degree, and they were making much less income per year. At age 32, almost half of them hadn't achieved school qualifications, and they were still making about $10,000 a year, less than typical readers. And they were overrepresented in the lower SES groups and underrepresented in the higher socioeconomic groups. At age 45, hot off the press, again, you've got those with dyslexia overrepresented in the lower SES categories and underrepresented in the higher economic groups. With regard to psychological problems, again, we found some differences with those with dyslexia having a greater likelihood of conduct disorder symptoms and anxiety, more likely to feel stress reactivity, feelings of alienation and aggression, and significantly more likely to be diagnosed with an anxiety disorder. So the bottom line is early identification and intervention is crucial in order to reduce the likelihood of these disorders, these outcomes. So in my first few years at the University of Auckland, some important things happened. One of them was that I was fortunate to join the Auckland Birthweight Collaborative longitudinal study founded by Professor Ed Mitchell. And I got involved in the study at about the age 7 assessment, and I've been mostly interested in early predictors of depression, ADHD, problem behavior, and IQ. And for example, here in a study led by Rebecca Sleckerman, we found that maternal stress during pregnancy is associated with moderate to severe depression in 11-year-old children, particularly if they're born small for gestational age. In another study led by John Thompson, we found that paracetamol taken during pregnancy increased the likelihood of symptoms of ADHD and problem behavior at ages 7 and 11 years. But these are adverse environments. How about genetics? I started getting interested in molecular genetics about the same time that the Dunedin study started to publish some really important candidate gene studies. So for example, they found a single nucleotide polymorphism on the serotonin transporter gene increased the likelihood of depression. And another study, they found that carriers of a COMT gene were more likely to develop schizophrenia if they smoked cannabis during adolescence. So those of us in the ABC study thought this was pretty cool, and so we jumped on the genetics bandwagon. We looked at the DAT1 transporter gene and its association with both ADHD and depression. We looked at the COMT gene, which regulates the catecholamines in the prefrontal cortex, and its link to ADHD and IQ, and the brain-derived neurotrophic factor gene and its association with IQ. But the important point here is that of gene-environment interactions. So for example, in that Dunedin study research I pointed out earlier, the probability of depression was only significant when there was a history of maltreatment was matched by one or both of the serotonin risk genes. And for us in the ABC study, we found that children born small for gestational age were more likely to have depression symptoms if they had a certain variant of the DAT1 gene. And again, for children born small for gestational age, we found a protective effect in this study. We found fewer ADHD symptoms if they were carriers of a certain DAT1 genotype. And finally, maternal smoking during pregnancy resulted in offspring with IQs on average 4 points less. And that in comparison to mothers who weren't smokers. And the effect more than doubled if they were MET carriers of the BDNF gene. So that's 8 IQ points, which is more than half a standard deviation below the mean of IQ. So, a second important thing happened in my first few years at the University of Auckland. I started teaching developmental psychology. And I stood up at the front of the class and I said that we're born with 100 billion brain cells and that's all we're going to get. I also said that it's really only in the first few years of life that stimulation and the environment can impact on the developing child. The rest of the time, the brain's kind of static. Well, we know that's not true, in part thanks to Sir Richard Fall's research on neurogenesis. We create new brain cells throughout the lifespan, and our brains continue to change and make new connections throughout adulthood. So what is neuroplasticity? It's the capacity of the brain to rewire through experience. And this figure shows how this rewiring can happen. So we're born, the brain overproduces both neurons and their connections. And then we go through a long period of pruning, of progressive synapse elimination, where only the strongest connections remain. And I think about the brain before pruning as being like a dense forest with tangled branches. Neuroplasticity allows the brain to be responsive to experiences. And I've already taken you through some examples of how this can be a negative thing, but it can also be a positive thing. And one of the most striking examples of this that I found are those who have had a hemispherectomy. So Mike Corbelis took a few of us along to meet 4 individuals in the Auckland region, 2 who had had a complete left hemispherectomy in infancy and 2 who had had a right hemispherectomy. And the important thing is that they were all living relatively independently, and you would not know to talk to them that they only had half a brain. Neuroplasticity also allows our brain to benefit from stimulation, so it can change from playing a musical instrument and through cognitive intervention. And Girgitsa for her PhD and Haimi for her master's asked the question, how about learning a new language? Can that change the brain? And what we found, this panel are monolinguals. These are bilinguals using their native language, and this is bilinguals using their second language, English. And what we found compared to monolinguals, a more extensive neural network in bilinguals when processing, they were doing lexical decisions in English and also their native language. So we found support for the idea that bilingualism can change the brain. There's also some research suggesting that there's a cognitive advantage to bilingualism. So switching between languages can enhance cognitive activation and processing. And we found a bit of support for this in an fMRI study with a bilingual woman who was dyslexic in both her languages. But to look at this further, I had my colleague Leda Tomic come from Brazil, and she spent a year in my lab. We did multiple EEG experiments, and we collected data from monolinguals, bilinguals, monolingual dyslexics, and bilingual dyslexics. And so watch this space. We're just analyzing the data now. And finally, a plastic brain can allow the brain to compensate when areas are not functioning optimally. So for example, in the case of dyslexia. And in our first few years as a lecturer, we had a group that won a Vice Chancellor's Development Fund Award, and it allowed us to go to Melbourne, to the Brain Research Institute, to do functional magnetic resonance imaging. And at that time, we did not have an fMRI scanner in New Zealand. And I was interested in language, and so typical readers, we found in the left hemisphere 2 areas of maximal activation, a more forward inferior frontal gyrus activity, and a more posterior left temporal cortex activation. And in dyslexia, we found a lack of that left temporal lobe activity. And it was a first for New Zealand, but internationally it wasn't a first. So this figure shows a review study showing 9 studies, all finding this lack of that posterior left hemisphere language activity during language tasks. But that's not what I really found interesting. What I found interesting was this increased activity in the right inferior frontal lobe. So the right hemisphere is trying to help out, it's compensating, but it's having a bit of a hard time because the right hemisphere isn't really specialized for language. It likes information presented simultaneously rather than sequentially. But a good result of this outcome, of this research, is that it did convince the Ministry of Education to finally formally recognize dyslexia as a real disorder in 2007. In 2009, we did another fMRI experiment, this time in Auckland, and we looked at shapes. We didn't find any group differences in nonverbal activity. We looked at different types of words. I wanted to kind of look at the orthographic areas of the brain and the phonological areas of the brain. And here, for dyslexia, again, we found this significant right hemisphere activation, significant as the cognitive and phonological task demands increased. So for the last fMRI experiment in 2012, we decided to focus on a phonological task. So our colleague Sally Shaywitz sent us a rhyming task. And this fMRI experiment was part of a larger Auckland comorbidity study that I did with Anna Wilson. And we did a fancy correlation technique thanks to Reese Roberts. And our aim was to determine the relationship between activation and reading performance. And here is our outcome. So what we found is a main effect of condition. We didn't find a group by condition interaction. So for all our participants, we found this significant left hemisphere activity in the inferior frontal cortex. But this is what's really interesting. As expected, for typical readers, as that activity increased, so did their task performance on a phoneme reversal task. In contrast, for those with dyslexia, as the activity in the left hemisphere increased, their performance decreased. So we also looked at activation outside of that traditional language network, and we found activity in the bilateral temporal lobes and in the insula. And in the left panels is the phoneme reversal task, and in the right, a word attack task. So they're sounding out nonsense words. And as expected, this compensatory activity wasn't really helping typical readers on the tasks, but it certainly was for those with dyslexia. And this is these areas here, the temporal lobe, and then the insula tucked in here with connections to the frontal lobe and the temporal lobe. So reading performance is mediated by a different network in people with dyslexia, possibly due to a genetic vulnerability to atypical left hemisphere wiring. But there's also some research suggesting that the corpus callosum is also atypical, and we found some suggestive research that this is true. We looked at interhemispheric transfer time, and for adults, it takes about 12 milliseconds for words to go across the corpus callosum. For individuals born without a corpus callosum, it takes over a minute because the information has to go through a different route. And for children with dyslexia, about 40 milliseconds, so significantly longer than adult typical readers. So watch this space, again, something I'd like to do in the future. So ADHD, most of the research has been focused on the prefrontal lobe with MRI showing reduced density and fMRI and PET studies showing decreased blood flow during executive functioning tasks such as these. And hopefully this looks familiar. I used a lexical decision task, and this involved decision making, and it produced a lot of frontal lobe activity in typical readers, so in the cingulate gyrus. But for those with ADHD, we don't see that activity. It's underactive because of neurotransmitter, dopamine and norepinephrine depletion. And that's one reason why stimulant drugs, such as dexamphetamine and Ritalin, work. They normalize the frontal cortex and increase the activation. ADHD is also one of the most highly heritable of our conditions. And again, in the ABC study, we found that maternal stress during pregnancy increases the risk of ADHD symptoms and poor behavior, and that effect doubled if they were met homozygotes for the COMP gene. And ADHD symptoms are also very stable, so only about 20% of them completely outgrow ADHD symptoms. But in the younger years, the symptoms of problem behavior are less stable. So behavioral problems can present as early as 2, but they typically don't persist. And Dr. Steph D'Souza gives parents hope who are in the midst of the terrible 2s. New directions. Also in 2004, I became involved in a new longitudinal study called Growing Up in New Zealand. And after a long gestation, in 2009, we had the antenatal data collection wave, where over 6,000 pregnant women and their partners were interviewed on a wide variety of constructs. And some of the early findings that have significant implications for health services are that depression symptoms are actually increased, they're more prevalent during pregnancy than during the postnatal months. In fact, it's quite rare to have women developing depression symptoms postnatally. And new fathers also get depression. And Lisa Underwood led all of this research. So there was numerous reasons why a new longitudinal study was needed in New Zealand. And our large sampling region allowed our cohort to be broadly generalizable to contemporary New Zealand. And our large sample size allows us to look at subgroups, gives us the statistical power to look at subgroups. And here with Liz Peterson, we are co-domain leaders for the psychosocial and cognitive domain. And finally, earlier this year, we were awarded a Marsden grant to study polygenic and environmental markers of mental health status in New Zealand children. And our aim is to identify the most significant biological and environmental pathways underpinning child mental health. And our hope is that one day our very alarming depression and suicide rates will be reduced. Thank you.